For those of you who I haven't had a chance to meet, my name is Tina Bose. I'm a scientific liaison at Adaptive Biotechnologies. I'm here today to speak to you a little bit about what we do and why we do it. So we're a biotech company that our headquarters are in Eastlake. I'm showing you a little picture of our building, and we're not too far away from where we spun out of, which is just down the street from the Hutch. And in 2009, our three founders, which are the researchers that were working at the Hutch and that are still working there now, found a really cool way to characterize or profile a patient's adaptive immune system. And they do this by sequencing the T cell receptors and B cell receptors. And don't let the jargon get to you. I'll explain that in one moment. And what I should just quickly introduce is B cells. You may not know what that does. Those are the cells in your body that make antibodies. And T cells, you may have heard of those as well. And not only can they help other cells do their job better, but sometimes they can directly kill infected cells. So killer T cells is another name you may be more familiar with. So I wanted to think about how can I explain the importance of your immune system. And I actually thought of this last night kind of late. And I'm wondering how many of you are familiar with this image. So hopefully some of you are familiar. So for those who are not familiar with it, this is a movie, not so good one. Bubble Boy, and although the movie wasn't that great, what it was a very good example of why your immune system is important. So this is based on a true story of a boy who was born with a rare genetic disease where this boy doesn't have any functional T or B cells. And so what that means is that he just can't defend himself from infections from just any run-of-the-mill bacteria or virus. And so he has to live within this sterile bubble so he can stay alive. So fortunately, most of us here, including me, have a functional adaptive immune system. So our B cells and T cells function properly. And you might ask, how is it that your B cells and T cells in your body can recognize everything that you will ever come across in your entire lifetime and do it in a very functional way? Well, they do this by these cell surface receptors that are present. And there's actually specific gene segments that recombine and rearrange to form this unique sequence of these receptors. And so the strategy for immunosequencing, the sequencing of T and B cell receptors, what Adaptive has done is to use this genetic code and sequence that in a way that we can track the different B cells and T cells. It just so happens when these genes recombine, the level of diversity that they generate is immense. Because just imagine, they have to be able to defend you from anything you'll ever come across. And just as an example, a certain type of T cell, just the number of large diversity that you have, there's 10 to the 15th possible combinations that are generated from this recombination event that takes place. And so that's 1,000,000 billion possible combinations. So you really need something like next-gen sequencing and bioinformatics to be able to really track this at a fine and high resolution. So what we do is we take T cells and B cells that are present in different tissues, we take up the DNA, we sequence that DNA, and then the sequence for these receptors can serve as an individual barcode or a fingerprint so that you can use that unique sequence to then track it over time. And you might ask, so what's the purpose of being able to sequence at this level? Well, we really want to change the course of medicine by improving patient care. So we're just at the cusp of understanding what a healthy adaptive immune system looks like. So now we can ask, how does your immune system change when you have a certain disease? And then use your immunosequencing data to serve as a biomarker for diagnosis of that disease. And also there's a couple of other ways to do that. You can look at the therapeutics. So there are a lot of drugs that are present out there, but not every drug works the same way in every person. So you need a way to stratify the different patients so that we can make sure that only the patients that will benefit will be exposed to that drug. So you could use a repertoire information for that. And finally, for monitoring an immune response, you could also use immunosequencing to do that. And I'll just go through this quickly. This is sort of going from sample to data. Patients, clinicians and researchers send us their samples. We take out the DNA, sequence it, process the data, and upload it into our cloud-based software, the analyzer. There they can analyze their data. They can compare their data against open source data sets. So it's a one-stop shop. And just to kind of finish off, in my 3 years, or a little under 3 years that I've been at Adaptive, any place where B cells and T cells could be playing a role, you could be using immunosequencing. So that could be allergy, autoimmunity, and a whole host of different diseases. But the area that's really just, it's hot and has really blown up is cancer research and immunotherapy. And being not that far away from the hutch, I'm sure you hear that on NPR all the time. And so that's the field that is really using immunosequencing really greatly. And that's it. So thank you for your time, and the organizer, I'll take questions. So the question was, the research or technology that we're working on, can it be used for vaccine design and autoimmunity like diabetes? And in fact, absolutely it can, and it is being used. So what I didn't discuss was that, you know, what our success, how we mark our success is really incorporation of the technology into various fields. And so the number of publications as well as the number of clinical trials that are incorporating our technology has really grown. And vaccine design, especially using vaccines to even cure cancer, is an open and active field. So both are being currently used even in this area. Yeah, so some of what they do is they are engineering T cells. So they track these components that they put into the body, right? They take out the T cells, they engineer it, they put it back in the body, and then they need to be able to track those T cells. So that's exactly what they could use it for. I'll take one in the back. No, this technology is not using CRISPR. However, researchers that are using CRISPR in different modes. I only talked and focused on human. We also have products that are focused on mouse research, so preclinical research, where they are using CRISPR and they want to track T cells, and so they'd be using them. So that was 3. Can we do more? I'll take one more. Let's talk about the cost afterwards. I learned my lesson. But, so basically our customers are academic researchers, they're clinicians that work with researchers, they're biotech and pharma companies that also have their projects. And the samples are pretty flexible. Our chemistry is very focused in the sense any sample that has B cells or T cells could be using this technology. So it's pretty wide, all different kinds of fields in medicine, both preclinical, so mouse research, or human research, so clinical trials.